Hello, my name is Anthony Zamora. I'm an assistant professor at the Medical College of Wisconsin. Today, I'm going to be sharing some of the work that we've been working on in cell-based therapies on the Beacon platform. The title of my talk is Leveraging Real-Time Biology to Streamline the Discovery, Validation, and Translation of Cell-Based Therapies for Cancer. So there's three main case studies that I'll be covering today. The first being on some of the developments we've worked on in the TCR T-cell space. The next case study will be focusing on determining the biological properties of CAR T-cells that favor tumor cell killing. And then briefly in the last case study, I'm actually going to present some more recent data where we're able to compare the different cancer immunotherapies like bispecific antibodies and CAR T-cells, and then look at the transcriptional profile of these cells in order to gain more insight into the different mechanisms of action of these different therapeutics. As a starting point, I'd like to give an overview of some of the factors that lead to effective anti-tumor responses. And so whenever we're thinking about developing a cell-based therapy for cancer, there's certain tumor-centric and immune-centric parameters that we have to take into consideration. In terms of the tumor-centric parameters, we want to focus on a target that's selectively present on tumor cells and absent on healthy cells. So in the schematic here, you'll see from right to left, there's overexpressed antigens that are going to be expressed on both cancer cells and healthy cells. And so even though you can target those with a therapeutic, there is a much greater likelihood of generating off-target toxicities. Now, if you go all the way to the left side of this figure, you'll see single nucleotide variants and fusion genes. And so these are mutations that are unique and only present in cancer cells and absent in healthy cells. And so we think these are good targets, from a therapeutic standpoint, in order to increase the tumor specificity and at the same time minimize any off-tumor toxicity. And so we want to make sure that we're focusing on these tumor antigens that differ from self-antigens. And so when we think about the immune system, the immune system is really keen at targeting, pathogenic antigens from like viruses, but it wants to at the same time minimize any any non-specific responses to self-antigens. And so we want to make sure that the tumor-centric parameters focusing on these antigens that differ from any self-antigens. And then ideally, we'd want to target driver mutations that are necessary for tumor cell survival. And so we think that this will decrease the likelihood of any relapse events from occurring later on. Now, switching gears to the immune-centric parameters. So there's various factors that we should take into consideration depending on the type of tumor and then the type of immune therapy being given to a patient. And so there's immune infiltration, meaning how well can the T cells get into the tumors. We want to make sure that we generate a robust response. And so by targeting multiple antigens, we actually minimize the likelihood of the tumor evolving and then ultimately evading immune recognition. And again, we want to balance specificity and tolerance. And so this is one of the main reasons we like to target these tumor neoantigens, because if you look at the figure here, they tend to be more distant from self and potentially more similar to a pathogenic antigen. And so we think that these are the ideal targets when we're thinking about developing therapeutics for patients with cancer. Now, throughout my talk, I'm going to be mentioning different types of engineered T cells. And so what I mean by this is that, you can introduce either a synthetic receptor to a T cell, or you can actually replace the endogenous T cell receptor that's found on your T cells with one that's specific for your cancer neoantigen. Now, the choice of either utilizing a TCR T cell based therapy or a CAR T cell based therapy takes many factors into consideration. One of them being is the antigen that's being targeted. And so in a lot of the liquid tumors, like leukemias and lymphomas, the antigen is actually presented on the surface of the tumor cell. And so you can utilize a CAR T cell therapy to target surface antigens. Conversely, TCR T cells tend to be better capable of targeting presented antigens. So these are intracellular antigens that are presented in the context of your major histocompatibility complex, or MHC, or HLA in humans. And so these tend to be, dependent on MHC restriction, whereas CAR T cells are independent of MHC restriction. In terms of the number of antigens that have been shown to be able to induce these different engineered T cells, typically you have sufficient engagement with one antigen by a TCR T cell to induce a response, and CAR T cells typically take less than 100. Now, the affinity of the receptor for the specific antigen varies, and CAR T cells tend to have a much higher affinity for their receptor. And then they also utilize, co-stimulatory domains that are part of the CAR T cell construct. And both of them have challenges when it comes to manufacturing. These therapeutics require about 2 weeks to manufacture. And so depending on, developing an either patient-specific or off-the-shelf therapeutic, there's additional considerations that need to be taken into consideration. Now, in our research program in the lab, we're really focused on exploring this tumor-immune interface. And so some of the work that I started in my postdoc at St. Jude Children's Research Hospital was really focused on developing this pipeline to discovery. And so essentially taking it from discovery, so where we can, identify unique tumor neoantigens that could be targeted by the immune system, and then moving towards validation, so ensuring that, the different computational algorithms that we utilize are actually producing functional T cells and also antigens that can be recognized by these T cells, so we can perform in vitro assays to validate some of the discovery steps early on in this cycle. And then ultimately, the goal is to then translate this information and develop a therapeutic that can be utilized in patients with cancer. So whether this be an off-the-shelf therapeutic like a CAR T cell, or a personalized TCR T cell therapeutic targeting these intercellular cancer neoantigens. And so our lab really focuses on each of these stages with the ultimate goal of developing a cell-based therapy, for patients with cancer. And so early on, we were thinking about different technologies and different platforms that would allow us to interrogate, how the immune system is responding to these cancer neoantigens. And so we brought in some technology, initially the Lightning platform. More currently, we've been running our workflows on the Beacon platform. And so this is a really novel platform offered by Bruker Cellular Analysis. It has multiple capabilities that you would typically find across different pieces of equipment in the lab. Some of the unique and characteristics that I want to highlight are the ability to sort cells based on fluorescent staining. So you can, utilize an antibody to a surface marker on your cell. You can use size, position, and morphology to selectively choose cells of interest and then add them onto the system. You can measure different types of readouts. So you can look at analyte secretion. So if your T cell is producing cytokines, and you're able to do this at single cell resolution, which really gives you a lot of granularity in terms of how a single T cell is behaving. And you can do single effector to target ratios or multiple effector to target ratios. So it really provides flexibility in the different types of assays you want to perform. Another aspect is it actually serves similar to an incubator where you can culture cells on chip in a closed, sterile environment, and you can continuously replenish your media and reagents to minimize any toxic buildup into the pens. And then ultimately, you can perform these assays and link each of these readouts, so whether it be phenotype, whether it be cytokine secretion, or whether it be cytotoxicity, these can all be linked back to the same pen, which is very similar to a well, get robust characterization of your cell-based therapeutic. So you can utilize a platform essentially to expand T cells, to validate or discover new T cell receptors, and then to validate your CAR T cells against tumor antigens that are expressed on the surface of your tumor cells. Another unique component of this platform is that it utilizes these OptoSelect chips. And as I mentioned previously, these chips have pens which serve similar to like a well on a 96 or a 384 well plate, where you add your cells of interest, you can cold culture your cell-based therapy with your tumor targets, and then monitor the responses of those therapeutics over time. Now, the way that it adds the cells into the pens is using this optoelectro positioning. And so it generates these light cages around your cells of interest based on whatever surface markers you're interested in interrogating. It adds these as single cells, and then you can do multiple rounds of this electro positioning in order to increase either the number of cells or the different parameters that you want to utilize in your assay. And so the first case study that I'm going to be describing today is actually a collaboration that we worked on with Dr. Paul Thomas at St. Jude Children's Research Hospital, where we're looking to validate an engineered T cell receptor or an engineered T cell that expresses a T cell receptor that's specific for a fusion gene that's found in patients with fibrolamellar carcinoma. And a lot of the work that was involved with this was actually led by one of the graduate students in the lab, Tanya. And the results from these studies were actually recently published in Cell Reports Medicine. So for anyone that's interested in gaining more insight into some of the results, I would encourage you to check out the publication. So just to give a broad overview of fibromyalgia carcinoma. So fibromyalgia carcinoma is a liver tumor that primarily affects adolescent and young adults, and it's typically around 1 to 5% of primary liver carcinomas. Patients actually have a very poor outcome, and there's no known cure for FLC. And so typically the 5-year overall survival is about 35 to 45%, and the relapse rate is upwards of 60%. The key tumorigenic event in FLC is a highly conserved gene fusion. And so this gene fusion, as I mentioned earlier, seems to be an ideal target for patients with FLC, primarily because the fusion is actually conserved, meaning that the amino acid sequence in about 90% of FLC patients is the same. And so we think that this would be a really ideal target for an off-the-shelf TCR T-cell based therapy sometime in the future. And so our hypothesis is that this fusion gene is able to elicit T-cell responses in patients with fibromyalgia carcinoma, and that this could actually serve as a target for TCR T-cell based cell-based therapies. And so to give an overview of the ways that we validated these engineered TCR T-cells on the Beacon platform, we went through this opto cell therapy workflow, and we wanted to look at both cytokine secretion, rate of cytotoxicity, and then also phenotype the cells in order to determine whether they were expressing either certain surface markers and whether they were secreting specific cytokines. And so this essentially works like a sandwich ELISA, where the capture bead will bind your protein. You come in with a secondary protein to that same secreted cytokine, and then those secondary antibodies will actually have a fluorescent marker that you could detect on the system. And so as a first step, we actually added 3 different cytokine capture beads for well-known cytokines that play, an anti-tumor role. Then we selectively penned in engineered TCR T cells. We added labeled target cells. So these are artificial antigen presenting cells that presented this fusion gene. And these were labeled with CFSE so that we can monitor the target cells over time. And then we perfused in a caspase reagent that allowed us to determine the exact moment that the tumor targets were being killed. And then, as I mentioned, we added our antibody cocktail, which allowed us to then quantify the cytokines that were being produced. And so we actually tested various TCR T-cell constructs in order to determine which of these engineered T-cells would have the greatest anti-tumor efficacy. And so if you look on the left side of this slide, you'll see these are time-lapse images that were collected on the Beacon every 30 minutes, and we could then monitor, once the TCR T-cell came in contact with the tumor cell, and then how quickly after immune synapse was formed did the TCR T-cell kill the tumor target. And what's interesting, and one thing I really want to highlight, is the fact that these different constructs, so if you look at the SJ1-4 versus the SJ1-12 in the middle, the rate of killing varied depending on the specific TCR T-cell construct that was used. And so on the top, you can see this caspase reagent fluoresce in the PE channel once it was cleaved, which is an indicator of cell death. And this was significantly delayed when we utilized the TCR T-cell SJ1-4, but was much quicker utilizing the TCR T-cell SJ1-12. And then we also had our negative control, where we had the fusion target that was expressed, but a mock transduced T-cell to show that there wasn't any non-specific background death of the target cells. Another unique feature is the fact that we can actually generate these videos. And so in real time, you can monitor the T-cell coming in contact with your tumor target and the exact moment that that tumor target gets killed. And so, in this case, it was an intermediate rate killing. And so you'll see that the tumor target begins to fluoresce yellow. And that's an indication that that TCR T-cell was able to effectively kill that tumor target in that pen. Similarly, when we introduce the SJ1-12 with a tumor target, you'll see it was able to kill that tumor target much quicker within the first couple hours of generating that immune synapse. And in the case where we had our negative control in this last video, you'll see that even though these cells generated immune synapse, so the tumor cell came in contact with the T cell, that T cell never killed the tumor, which is due to the fact that it doesn't express a T cell receptor that can recognize that antigen that's being presented in the context of MHC. And so these were really exciting studies that really highlighted the capabilities of the Beacon platform, and again, give us a gauge of being able to determine at single cell resolution which of these constructs was mediating the most beneficial effect in terms of being able to kill the tumor. And in these bar graphs, as I mentioned earlier, we also added cytokine capture beads, and so we can additionally determine which cytokines were produced by the different constructs. And then we also had control target cells that expressed a wild type version of the protein. And so in the case of the SJ1-12, even though that cell killed much quicker than the SJ1-4, there was also greater off-target cross-reactivity. And so the fact that this is responding against a wild type protein is potentially of concern because it could be expressed by healthy cells, and then we would run into the scenario where we would have, off-tumor toxicity. And so we felt very strongly that the SJ1-4 was a much better TCR-T cell that we could then translate to the clinic and utilize in patients that were expressing this fusion gene. And similarly, when you look at the different cytokines that are being produced, the SJ1-4 had much greater polyfunctionality, meaning it was producing many more cytokines. So the triple cytokines are highlighted here in dark blue. And you can see there was a much greater polyfunctionality in this construct than in the SJ1-12. And so in conclusion for this initial case study, I just wanted to highlight that, we can induce these endogenous CD8 T cells in these patients to recognize these fusion genes. And this fusion gene was, targeted and was able to expand CD8 T cells. We found that the SJ1-4 and SJ1-12 TCR T cells were both targeting the fusion, but they had differences in terms of their rate of cytotoxicity and the rate of polyfunction, or the amount of different cytokines that were being produced. And so SJ1-12, even though it killed the tumor targets much more quickly, these engineered T cells actually displayed a much higher level of cross-reactivity against wild-type peptides, whereas the SJ1-4 displayed polyfunctionality, so it was a triple cytokine producer, and it also had much reduced cross-reactivity to the wild-type protein. So again, suggesting that this might be the more beneficial cell-based therapy to introduce into a patient. And so switching gears to our second case study, this study was actually led by a graduate student in the lab, Alfredo Colina, in collaboration with Dr. Narav Shah, who's the clinician who's actually giving these CAR T cells to patients, and then Dr. Brian Johnson, who was the former director of the cell therapy lab here at the Medical College of Wisconsin. And so in case study 2, we're really looking at trying to identify the biological properties of these bispecific anti-CD19, CD20 CAR T cells that favor tumor cell killing. And so another area of research in the lab focuses on ways that we can improve the in vivo persistence and clinical efficacy of these dual CAR T cells by assessing the long-term outcomes in patients with non-Hodgkin's lymphoma and leukemia, and then also determining whether differences in manufacturing, so if you generate your CAR T cells using different cytokines, what is the end result? Will you generate a more functional CAR T cell, or could you potentially drive the CAR T cell down a program of terminal exhaustion, so by the time you infuse it into the patient, there's going to be significantly reduced functionality? And so in these studies, that's exactly what we did. Initially, we were comparing CAR T cells that were generated from the same patient using IL-2 versus IL-7, IL-15. And then as a follow-up study, we then determined whether we could identify immune signatures or biological characteristics of CAR T cells that correlated with clinical outcome. And so similar to the workflow that I described earlier, we utilized the ability to add specific cytokine capture beads. And these were actually chosen by characterizing these single CAR T cells on an isoplexis assay, where we looked at multiple analytes and then selected those that had the biggest difference between the CAR T cells that were manufactured under IL-2 or those that were manufactured under IL-7 and IL-15. Now, as the next step, we added the CAR T cells to our pens using the optoelectrode positioning. Again, we utilized tumor targets that were labeled with CFSC. And then we perfused in media that contained this caspase reagent so that we can monitor the exact moment that the tumor targets were killed so that we could determine the rate of cytotoxicity. And then we added our antibodies to look at surface marker expression, and then also, again, to serve as a secondary antibody to cytokines in order to perform similar to a sandwich ELISA, determining the amount of cytokine that was being produced. And similarly, as I mentioned earlier, you can utilize these pens to select specific targets in the channels and then, get single cells into each of the pens in order to characterize single CAR T cell function against single target cells. And so what was interesting is when we looked at the different CAR T cells that were manufactured under IL-2 or under IL-7 and IL-15, we saw that they were very polyfunctional, meaning that they were able to kill and they were also producing multiple cytokines. But what was different was we saw that the rate of killing and also the extent of cytokine production varied depending on whether IL-2 was used during manufacturing or whether IL-7 and IL-15 was used. And so in these plots, what you can see is that, across all quadrants, there were cells that were either producing, interferon gamma in this specific case, and that were killing very quickly. So, reduction in time before we saw this cytotoxicity. And then also some that were producing high amounts of interferon gamma, but had much more delayed killing. And then there were also cells that produced very little interferon gamma, but killed quickly, and those that were slow killers and produced low amounts of interferon gamma. And so if you compare then in the pie charts to the right, the amount of polyfunctionality of IL-2 versus IL-7 and IL-15 primed CAR T cells, and if you also look at the frequency of non-functional CAR T cells, what you'll see is that the IL-7 and IL-15 primed CAR T cells had a much higher polyfunctionality and had a much lower frequency of non-functional CAR T cells. Again, suggesting that, IL-7 and IL-15 is more beneficial during manufacturing if the result is, or the desired result is to increase the cytotoxicity and cytokine secretion by your CAR T cells. We also looked, as I mentioned earlier, at differences in responders versus non-responders in patients that receive CAR T cell therapies. And again, using time-lapse images, we can monitor the exact moment that immune synapse was formed between our immune cells and our target cells. And then after immune synapse is formed, how quickly are the CAR T cells killing their tumor targets? And so you can do this over multiple channels. So you have bright field images, you have different fluorescent channels. In this case, we're using FITC and PE. But there's also the ability to interrogate in the DAPI channel, in the APC channel. And so it really gives you the ability to look at multi-parameter assays and then characterize different cytokines, different surface markers, and then also, as I mentioned, the rate of cytotoxicity. And so going from left to right, as you can see, is that this immune synapse was formed very early at the start of the assay, but even though these CAR T cells all contain the same CAR construct, they differed in their rate of killing. So there were some that, killed very quickly and others that killed very slowly. And so we think that there's some underlying biology that really drives these differential responses in patients that are administered CAR T cells. And so again, you can actually monitor this in real time by playing these videos. And so you'll see there's immune synapse formed between the tumor target and the CAR T cell. And this CAR T cell killed within the first 2 hours of starting the assay and the immune synapse being formed, whereas the one on the right here had significantly delayed killing and actually took much longer to kill the tumor target. So even though immune synapse was formed immediately, it wasn't until 8 hours later that CAR T cell was actually able to kill the tumor target. And so when we, in some of our initial studies, when we started comparing, which of these parameters seem to correlate strongest with, clinical outcome in these patients that were administered CAR T cells, what we started to see is that these CAR T cells that had a higher frequency of what we were calling fast-killing CAR T cells tended to correlate with clinical outcome, meaning that these patients were responders at day 28 post administration of the CAR T cells, And then in follow-up studies where we looked at, day 90 plus, those patients were still responders. And so we set up the assay so that we had control target cells that didn't express our CD19 and CD20 antigen to ensure that there weren't any non-specific killing of our tumor targets. And so if you look from left to right, what you'll see is that there is variability in the frequency of fast, medium, slow killers between patients, but all of the patients that were responders, which is indicated by the R at the bottom of these sunburst plots, tended to have a greater frequency of these fast-killing CAR T cells. Now, one thing that was rather surprising was if you look at the most outer circle, there was differences in terms of the different cytokines that were being produced. Now, if the CAR T cells produced 1, 2, or 3 cytokines, didn't seem to have as strong of a correlation with clinical outcome. But what was interesting in the one patient that was a non-responder in this initial cohort, we saw that it had a much lower overall rate of killing. So there were many more CAR T cells that were non-killers and a much lower frequency of fast, medium, and slow killers. Again, suggesting that rate of killing seems to correlate well with clinical outcome. And so in the second case study, really what we'd like to highlight is that, the bispecific CAR T cells across patients are very heterogeneous in terms of their functional responses. We also noticed that different manufacturing conditions can alter the functional responses. And so this is something that's really important to consider when thinking about, developing new manufacturing conditions to generate CAR T cells, and then looking for ways to standardize this across different institutions. And as I mentioned, CAR T cells that were manufactured under IL-7 and IL-15 displayed much higher polyfunctionality, and our functional data suggests that there was a trend in rate of cytotoxicity killing and clinical response in patients that were administered CAR T cells. And so the data that was collected from these beacon runs can also be used to parameterize steps in CAR T cell signaling and then model functional responses utilizing different computational models. And so those are additional studies that we'll be performing in the lab in the near future. And the last case study that I wanted to highlight is a study where we were actually comparing the ability of different cancer immunotherapies, in this case, BiTEs and CAR T cells, and trying to distinguish whether one therapeutic was generating more productive T cell responses compared to the other. And so these studies were led again by Alfredo Colina, one of the graduate students in the lab, and then Anand George, who was a high school intern that actually worked with Alfredo and myself in order to perform these assays. And so he had a summer research project and is continuing to perform these studies in the lab, where he's again determining differences in the T cell responses utilizing either BiTEs or chimeric antigen receptor T cells. And so both of these therapeutics have been utilized across various hematological malignancies and shown pretty good clinical efficacy. But they are different in terms of their mode of action. And so BiTEs, these are bispecific antibodies that have 2 linked single chain variable fragments from 2 different antibodies, and they can target the CD3, so this is a complex of the T cell receptor on T cells, and they utilize the other fragment to then target a surface antigen that's present on your malignant tumor cells. This is different than the scenario of a chimeric antigen receptor, because this is a synthetic receptor that's then engineered into a T cell, and it's comprised of an extracellular antigen recognition domain that's fused to the CD3 zeta chain for intracellular signaling, and it typically contains 1 or more costimulatory domains in order to provide signal 2 to ensure that you get high levels of activation and expansion of your CAR T cells. And so the research question here was, how does activation of T cells through either your natural T cell receptors using these BiTEs versus through synthetic chimeric antigen receptors alter the function of different T cell populations in the setting of cancer? And so this assay is similar to the 2 that I described earlier, but the main difference being that rather than using tumor targets, we utilize antigen presenting beads that express or that presented the CD19 on the surface of the antigen presenting bead. And then we were able to perform these assays over 24 hours. And then one step that we added here was that we wanted to determine whether there was correlations between the functional readouts in terms of the cytokines that were being produced, and then the transcriptional signature of the T cells that were activated with BiTEs versus those that were activated through the chimeric antigen receptor. And so we utilize OptoSeq on the Beacon platform in order to capture messenger RNA that's then present after lysing our effector cells. And then we can perform downstream transcriptional analysis to determine whether there's a unique immune signature between either the T cells that were activated with BiTEs versus those that were activated with CAR T cells. I want to highlight on the bottom here, looking at these time-lapse images, one thing that really stood out to us was that when we were comparing the engagement of our CAR T cells with these antigen-presenting beads, we saw that the CAR T cells tended to stay engaged with the beads much longer, and they would actually drag the beads around the pens, whereas the T cells, they had BiTEs, which brought the T cells to the interface of the antigen-presenting bead, tended to have much higher off and on rates compared to the CAR T cells that were constantly engaged. And so after characterizing the different cytokines that were being produced, we then utilize this OptoSeq 3' messenger RNA library kit in order to look at the transcriptional signature of these CAR T cells versus the T cells activated with BiTEs. And so this workflow is very unique in that you can actually flow in different fluorescent probes that bind to this nanopen barcode region on the oligo that's bound to the bead that you add into each pen. And so it gives you a fluorescent signal that you can then link to your nucleotide signal after sequencing and determine the exact pen that transcriptional signature correlates with. And so again, one of the real strengths of the Beacon platform is that you can link different functional readouts with different transcriptional readouts and get a more comprehensive assessment of what biology of the cells is actually driving these differential responses. And so after performing this assay, we performed dimensionality reduction on our sequencing data. And what was interesting is that we found that the CAR T cells tended to cluster in 2 separate clusters, whereas the T cells that were activated with the BiTEs had one very large cluster. And so when we looked at the different genes that were up or down regulated in these 2 different populations, what we saw is that a lot of the effector genes, so TNF alpha, so the cytokines, different cytotoxic granules like granzyme B, were much highly expressed or upregulated in our CAR T cells, where we saw a much lower upregulation of these genes in our T cells that were activated with BiTEs. And we also then determined whether there was concordance between these transcriptional assays and in the cytokine secretion assays. And what we found is that in certain cases, such as that with TNF alpha, there was a high amount of cytokine that was being produced, which also correlated with the amount of TNF alpha transcript that was present in our CAR T cells. And very similar to what we highlighted in transcriptional profiling, the specific BiTEs that engage the target didn't secrete high levels of TNF alpha or interferon gamma versus the CAR T cells, again, suggesting that both at the transcriptional level and at the protein level, there were differences in the chimeric antigen receptors that were activated with these beads versus the T cells that were brought into close proximity of the beads and activated with these BiTEs. And so in conclusion, I want to highlight that the Beacon platform allowed us to directly compare different immunotherapy regimens and monitor T cell responses based on either BiTEs or chimeric antigen receptors. And what was interesting is that the CAR T cells demonstrated this prolonged target engagement compared to BiTEs. And they also displayed greater polyfunctionality based on their upregulation of activation markers and production of effector cytokines. The increased contact time between the CAR T cells and targets could actually help explain why there were differences in the functional responses. So the longer immune synapse that was formed between the CARs and the beads likely induced a much greater activation of those CAR T cells. Now, one thing to consider is that while the CAR T cells displayed greater functionality, this could also be detrimental in a patient that has cancer, because we know that an overactive CAR T cell can, 1, either decrease persistence, 2, increase the exhaustion profile of those CAR T cells, but then also 3, can increase the amount of toxicity, so the detrimental effects that are resultant from these CAR T cells being highly functional. And so really we want to try to balance this activation and make sure that we're getting productive responses against the tumor targets, so maybe preferentially having more cytotoxic cells and fewer of those that are producing pro-inflammatory cytokines. And so I want to acknowledge, the various members in the lab that have contributed to these studies. I highlighted some of the work that Tanya and Alfredo did early on, along with some of the work that Anand did. But a lot of these experiments were actually supported by other members in the lab. So Dr. Ravi Shah is a research scientist in the lab. And then Jessica, she's one of the research technicians in the lab, along with Marlene. Both of them have really helped develop some of these workflows on the Beacon and carry out some of the experiments. And then lastly, I want to highlight, these studies wouldn't have been possible without the contributions of Dr. Nirav Shah and Dr. Brian Johnson, who provided the CAR T cells for the different CAR T cell studies that we highlighted today. And so with that, I'm happy to take any questions from the audience. Thank you.